Yes, good morning to all of you. So let us begin today's online class. So yesterday we had started a photosynthesis chapter, just introduction part we started. So today we shall continue that class. I'll just share my slide. Yes, I hope slide కనపడతా ఉంది అందరికి. Anybody respond? స్లైడ్ కనిపిస్తుంది కదా? ఎవరైనా అన్మ్యూట్ చేసి మాట్లాడండి.
Yes, sir. Conversation, sir.
Yes. Okay, maybe your friends they may join. Okay, let us start. So photosynthesis, what is photosynthesis? You can say it is an anabolic reductive carbonic process which is taking place in plants in a cell organelle that is called as chloroplast. So anabolic in the sense it is a constructive process where the simple molecules are they combine to form a complex molecule. So here simple molecules like carbon dioxide, water, the presence of light and chlorophyll color pigment, they are forming a complex molecule that is carbohydrate, sugar molecule that is glucose, C6H12O6. Along with this, oxygen is evolved and water is released. So it is a constructive process where simple molecules they are combined to complex molecule, as well as it is a reductive process. Okay, so it is undergoing a reduction. So water is being splitted. Okay, that hydrogen which is coming out from water, it is being accepted by a proton acceptor in the form of NADP. So it is a reductive process and as well as it is a enterogonic process where energy is utilized from external energy is utilized for this process to take place. That external energy is ATP and NADH. Okay. So these external energy is utilized in the further process of this total, that is carbon dioxide fixation. At that time this energy is utilized. So if you look at the photosynthesis equation, it is carbon dioxide C6H12O6 combines with 12 molecules of water in the presence of light and chlorophyll to give rise to C6H12O6 glucose molecule along with 6 molecules of oxygen and 6 molecules of water. So we discussed about the just brief history part yesterday. I won't go into detail. So the basis of photosynthesis, we can say all plants are photosynthetic autotrophs. Autotrophs means they are preparing their own food. They don't depend on any other or host for its food. So it is preparing food photosynthetically processed. That is why these are known as autotrophs. And there are few bacteria and protists also that which perform photosynthesis and they come under this category. So autotrophs generate their own organic matter through photosynthesis. And sunlight energy is transformed to energy stored in the form of chemical bonds. So here nothing but the light energy is converted into kinetic energy. So here you can see, so light energy is harvested by plants, the other photosynthetic autotrophs where the so here sunlight is the source of light. So as we said that this whole photosynthesis is taking place in a cell organelle called chloroplast, which is present in the leaves. Okay, in the leaves, this chloroplast is present where the photosynthesis is occurring, where carbon dioxide, water, and light energy is trapped and it is converted into glucose carbohydrate. The oxygen is released. So, oxygen is released and even water is released. So, why are plants green? So, plants are normally green color. Why? So, because if you see the plant cell, it consists of number of green color like structure cell organelles which are known as uh, chloroplast. Chloroplasts are the color pigments which are green in color. Okay? Because of the presence of a color pigment called chlorophyll. So here you can see further if one chloroplast is enlarged. So this is how it looks. So chloroplast is the largest cell organelle present inside a plant cell. So the thylakoid membranes of the chloroplast is impregnated by photosynthetic pigments, that is chlorophylls and carotenoids. So these chlorophylls are green color pigments which are present inside the thylakoid region of the chloroplast. Yeah. 1 second, just 1 second. Yes, sorry for the interruption. Yeah. So I hope I am audible. వాయిస్ వినిపిస్తా ఉంది కదా అందరికి?
Yes, sir.
Yeah. So here the this is the chloroplast where you will be finding the uh, color pigment. So as I said earlier that today I said you that there are there is a category of organelle that is called plastids present inside the plant cells. These plastids are sac-like structures, double membrane structures which store the different material inside them. So depending on the type of material, they may be termed as leucoplast, then chromoplast, then chloroplast, and so on. So the first one, leucoplast, are the sac-like structures of plastids that store the food material inside them. So food material is stored. So that may be proteins, carbohydrates, fats, oils, anything. Okay. So that is first one. Second one is the chromoplast, which store color pigments. Okay. In that carotenoids, xanthophylls, phycobilins, and so on, which store different types of color pigments inside them: yellow, orange, red, brown, so on. So that is second chromatoplast. Third one is the chloroplast. So this is chloroplast. Okay, this is the third one. It stores green color pigment inside them, which is called chlorophyll, and it is green in color. Okay, this chloroplast actually performs the photosynthesis. So this is the main organelle responsible for the photosynthesis process. So if you look at the overview of photosynthesis, so photosynthesis is a process which autotrophic organisms use sunlight, sun as for the light energy to make sugars and oxygen gas from carbon dioxide and water. These is carbon dioxide, water, light energy used the presence of color pigment called chlorophyll and glucose and oxygen evolved. So this is another picture which is showing here. So these, when you take a cross section of these, so as you know, this is the upper epidermis, this is the lower epidermis. So inside this, so this is the palisade tissue, parenchyma tissue, and this one is the spongy parenchyma tissue. Okay, so spongy parenchyma tissue has more air space compared to the palisade tissue. So in both the tissues, we'll be finding green chloroplast structures. So here you can see. So this region is known as mesophyll. That is upper epidermis and lower epidermis. Between region is known as mesophyll, which is comprising of two layers of tissues. One is palisade tissue and spongy tissue. So here, in both the type of tissues, you will be finding these green chloroplasts present. Green dot structures are the chloroplasts. And here in the lower epidermis, there are stomatas also present, which are responsible for what? For exchange of gases, this carbon dioxide entry and oxygen release. So there, this exchange of gases is taking place by these stomatas. Okay. So if you look at the structure here, chloroplast structure, so it has outer membrane, has inner membrane. There is a space in between which is known as intermembrane space or periplastidial space, you can say. So outer membrane is permeable compared to inner membrane, which is not permeable. The less permeable, you can say, outer membrane is more permeable. Which somebody is repeating here. So here the outer membrane is more permeable and inner membrane is less permeable, and the intermembrane periplastidial space is present. So along with that, we have what? We have uh, the lower side is filled with a fluid which is comprising of photosynthetic pigments that is known as stroma. Jelly fluid is there, which is known as stroma. And there are sac-like structures which are present here. So these sac-like structures are known as grana. Okay, so these sac-like structures are known as grana. Whereas if they are present in bundle, okay, like this, so one above the other, if they are present in a stack of coins, you can say, just like stack of coins, so that is known as granum, like this. So single is known as grana, and by bundle it is grana. There are few extensions here which are connecting all these bundles. Okay, so this extensions between one granum and another granum in the stroma are known as stroma lamellae or thylakoid compartment. So this is thylakoid compartment and so this is thylakoid. These extensions are known as what? Stroma lamellae. So here this picture is another picture. This is a electron microscope picture of the chloroplast. So this is the basic structure of chloroplast. You can say. Okay, here. So here one thing you remember that the photosynthesis has two phase of reactions. Seven, the light phase and dark phase. So light phase is taking place in presence of light, which takes place in the thylakoid region. Okay? So the thylakoid region takes place, and inside that, the stroma region, the dark phase takes place. So before going into the reaction part, we discussed about the photosynthetic pigments that are present, that are used in the photosynthesis. So what are they? So the photosynthetic pigment can be found in thylakoid and intergranal lamellae. So the functions of this pigment is to absorb the visible light. So one thing you remember that whenever the energy, okay, the sunlight from where the light energy is utilized. So these light energy, it comprises of different rays. Okay, so we are able to see the visible light, which is in the range of between 380 nanometers to 790 nanometers or 800 nanometers. So there are other rays like the gamma rays, X-rays, V rays, infrared rays, microwaves and radio waves, which are not visible. Okay, so they are also being radiated by sun. So we are able to see only visible light. So photosynthesis utilize the visible light. They absorb the visible light, which is in the range of 380 to 390. So photosynthetic pigments are the substance that absorb visible light within this wavelength. So each wavelength is different, having colored light. So as you know, the visible light has the range colors, the range of seven colors are there. That is with your so violet, indigo, blue, yellow, orange, red. The same violet, indigo, blue, green, yellow, orange, and red. Okay. So how many chloroplast pigments are there are they taking part in the photosynthesis means? So the main pigments are chlorophyll A, chlorophyll B. Even there are chlorophyll C, D, E also. And the other pigments are carotenoids, xanthophylls, and phycobilins. Okay. So photosynthetic pigments, if you see, there are two categories. One is the principal photosynthetic pigment, another one is the accessory photosynthetic pigment. In plants, chlorophyll A is the primary photosynthetic pigment, you can say. Surrounding this chlorophyll A, there are other accessory pigments surrounding it. So all those accessory pigments are what? They are chlorophyll B, C, D. There are carotenoids, again, which are carotenes, xanthophylls. Okay. Then other category is phycobilins, where phycoerythrin is there, phycocyanin. So chlorophyll A is the main pigment, whereas in microorganisms like bacteria, with photosynthetic bacteria, there the primary pigment is bacterial chlorophyll, which is present. Okay. So the difference between chlorophyll A and B is uh, if this is the structure, you can say the chlorophyll pigment A and B both are same structured, where it has a porphyrin head and a phytol tail region comprising 20 carbon atoms sterified with the third payroll ring. So these uh, chlorophyll A and B structures have a central magnesium atom surrounded by four pyrrole rings. Okay, one, two, three, four. So the 20 carbon alcohol and tail phytol tail region is esterified and esterified with the fourth pyrrole ring, whereas the main difference between C chlorophyll A and B is in here in the second pyrrole ring at the third carbon atom region where a methyl group is present in case of chlorophyll A and an aldehyde group is present in case of B. So that is the only difference we can say in chlorophyll A and chlorophyll B structure where the ethyl group is present in second pyrrole ring. So this is the tail region, both the case. Okay, methyl tail, and this is the head region having four phosphoric head region having four pyrrole rings. Okay, so one, two, this is the third one, this is the fourth. So to the fourth is alpha ring attached. So coming down to beta, so carotenoids. So here carotenoids are there, which comprises of carotenes, xanthophylls, and so on. Even say zeaxanthin. So the other photosynthetic accessory pigments you will find. Okay. Then coming down to the summary of photosynthesis, if you say what is total occur. So it occurs, photosynthesis occurs in many kinds of bacteria. They, so only uh, chloroplasts are able to carry out the photosynthesis. Basically, chloroplasts are the cell organs where photosynthesis occurs. Water and carbon dioxide are two raw materials that are required for photosynthesis. The presence of light and chlorophyll color pigment. Then photosynthesis takes place in three stages. First stage is the capturing of light energy, solar energy, coming from sunlight. So membrane system that is the grana and stromal annulate, they trap the light energy as they have the pigments like chlorophyll, carotenoids, phycobilin present embedded inside them. Then they use the solar energy to make what? Make energy in the form of ATP, NADPH. So NADPH they are the reducing point. Mainly ATPs are produced here during the light phase. And second part is during the dark phase, the photosynthesis, they use these ATPs and NADPH that are produced during the light phase to synthesize organic molecules from carbon dioxide. That is the actual carbon dioxide fixation process occurs here. The second, that is the dark phase. So that takes place in stroma as it has all the enzymes required for reduction of carbon dioxide to carbohydrates. Ultimately, the end product is carbohydrate 6H12O6. So these are the steps that are involved in photosynthesis. So here you can see the color of light is not the color that, that is absorbed. Okay? So chloroplast absorbs light energy, they convert it to chemical energy. So here when the light falls, chloroplast, so the green light is reflected. Okay? The remaining all lights are absorbed. That is why we see green color over the plant parts, that is the. So the mechanism of photosynthesis, you can say it is divided into two phases. First one is the light phase, and the second one is the dark phase. Light phase and dark phase. So light phase, what all occurs here is photolysis of water is there, phosphorylation is taking place, and you can say that in mainly here light phase oxygen is involved, photolysis is taking place. So you can say that whatever oxygen is released during photosynthesis, this oxygen is coming from water, right? Breakdown of water in the presence of light. That's why it is known as photolysis of water. Then you can say that this is oxygen is coming from water, but not carbon dioxide. As well as light reaction, there are two photosystems, PS1 and PS2. Okay? So in these two photosystems, the light energy is converted into energy that is in the form of ATP. So by a process that is photophosphorylation. So ATPs in the light phase, the ATPs and NADH are produced. And during the so that is light phase. During the dark phase, what is happening here? So whatever ATP and NADPH are produced, they are utilized here for the carbon fixation. So that is taking place through a cycle called Calvin cycle, or it is also called C3 cycle. It was proposed by Melvin Calvin. So light reaction is the light dependent reaction takes place in where the grana of chloroplast and its products are NADP and ATP as we had said earlier. And in dark reaction is light independent and occurs in stroma of the chloroplast where the uh, CO2 fixation occurs. Okay, and it is converted into carbohydrate, that is glucose. It is the end product. So this is the reaction mechanism of photosynthesis. Now we will be studying in detail one by one what all the phases are there. So for discussing that, let us take the first phase, that is the light reaction phase, where it can be studied under five subheadings. The Hill reaction. Second one is the Emerson enhancement effect. Third is the photosystem 1 and photosystem 2. Then the electron transport and the proton translocation. And fifth one is the photophosphorylation, where cyclic and non-cyclic photophosphorylation is there that we will be dealing. So the first one is the Hill reaction. Okay? So this is the uh, first step in the light reaction phase, where Robert Hill, a biochemist from Cambridge University in the year 1937, worked on isolated chloroplast instead of that plant chloroplast. He demonstrated that isolated chloroplast could release oxygen in the presence of sunlight and suitable hydrogen acceptor such as potassium pericarboxylate, or you can say here DCPIP, which is dichlorophenol doping. So it is called popularly known as what Hill oxidant or Hill reagent. So in plants, NADP plus acts as the hydrogen acceptor and water acts as the hydrogen donor. So the evolution of oxygen during photosynthesis can be represented by the following equation. Here, so what is that equation? So here, this is the this is the equation where water. So here, Hill reaction, what he did, where Robert Hill, he conducted in a artificial way, where he used the water was acting as the hydrogen and the reagent A is acting as the hydrogen. So that A is nothing but DCPI. Okay. So in the presence of light, the chloroplast, water in the presence of a hydrogen acceptor, that is DCPI, it is also will reagent which acts as the hydrogen acceptor water. So hydrogen gets bind to this reagent and the oxygen is released. So the reaction what is saying that oxygen is coming from breakdown of water in presence of light. Okay? So this by this he could say that the oxygen is coming from water but not from carbon dioxide. So this is one. Second thing is Emerson's enhancement effect. So here Robert Emerson's enhancement effect is one aluminum red drop. So uh, Robert Emerson in 1950 from University of Illinois, USA, observed in chlorella a unicellular green algae, that is chlorella, used for his experiment, that red light of wavelength longer than 680 nm lowers rate of photosynthesis. This decrease in the rate of photosynthesis at higher wavelength of red light is called as red draw. Subsequently, Emerson and his associates observed that when both shorter wavelength light, that is 650 to 680 nm, and a longer wavelength light above 680 nm wavelength were simultaneously used, what they observed was the rate of photosynthesis was higher than the sum of the rates with separate beams. This phenomenon was came to be known as Emerson's enhancement effect. So, so the red drop was the quantum Lie drops dramatically as the far red region of chlorophyll region greater than 680 and was used. At that time, the photosynthesis rate drastically dropped. That is known as the red drop effects indicating the far red light alone. Photosynthesis. Then what they did is Robert Emerson and his associates they conducted another experiment where the rate of photosynthesis red and far red light are given together. Okay, so that sum was when both the lights were used, the photosynthetic rate increased drastically, and which is known as Emerson's enhancement effect. So enhancement effect showed that there are two photochemical complexes known as PS1 photosystem 1 and 2, PS1 and PS2, light reaction of photosynthesis that are linked by electron transport chain. So what Emerson said that there are two types of wavelengths that are taking place here. Depending on that, we named them as PS1 and uh, PS2. Okay, PS2. So one was PS1, which was using wavelength which is greater than 680 nm, and PS2 was the another photosystem that is using wavelength of light that was less than 680 nm. So here you can see red drop, so far red light alone. That is when the light is going beyond here around 680 nm, what is happening here? The photosynthetic rate is coming down drastically. So this region where the actual red light is there, so whenever it is coming here, when it is going beyond 680 nm, what is happening here? The photosynthetic rate is drastically coming down. So that is why this is known as red drop. Whereas when the light lower than 680 nm and the above 680 nm were used simultaneously, when both the lights were used, so what was happening here? The photosynthetic rate increased drastically. Okay. When they were used singly, photosynthetic rate was less. But when they both the lights were used, that is beyond 6080 nm and below 680 nm, the photosynthetic output was very high. So this was known as Robert Emerson's enhancement effect, and that could come out with the conclusion that there are two photosystems involved in the photosynthesis process. So, what are those photosystems? If you see, so here, this picture, you can see this is photosystem 1, and here is photosystem 2. So, the photosystem 1, it is utilizing, so let us discuss this. Photosystem 2, it is utilizing the light, sunlight, which is coming from sun, uh, which is below 680 nm, okay, of wavelength. So here, if you see in this picture, this region is the reaction center. It is known as E6AG. So here it is chlorophyll A, and all these pigments are accessory pigments. So in the center, this is chlorophyll A, which is the reaction center. Surrounding it, all the accessory pigments like chlorophyll B, carotenoids, phycobilins, and so on, they surround the chlorophyll A. What they do is they absorb these accessory pigments. What they do, they absorb the light energy in the form of photons, and they transfer the light energy to the reaction center. So reaction center, what happens here? It gets excited. Okay, when it gets excited, it releases electrons. So when it is getting excited, it releases electron to the primary electron acceptor. So here, the primary electron acceptor, case of PS1 is pheophytane. Okay, so pheophytane accepts the electron from where, and this gets excited. Okay, so this gets excited because it is losing electrons. So this electron, while it is passing, so you can remember here that this thing is the thylakoid membrane. All this thing is happened in the thylakoid membrane region, where this is the stroma part and this is the lumen part. Okay. So when the electrons they pass from one carrier to another carrier, what is happening here? They lose energy. Okay. So first point here from photosystem 1, the light enters, light energy is passed, it reaches the reaction center from accessory pigment. The accessory pigment lose the electron. This electron is accepted by primary electron acceptor pheophytane. Again, it is passed on to plastophenone pool, where plastophenone pool it further uh, transform the electron to cytochrome B, cytochrome BF complex, which is iron-rich complex, iron-rich protein complex. From there, the electron reaches the copper-rich plastocyan proteins carrier electron carriers, where the electron is ultimately reached to S1. So PS1 here, the reaction center is P700. So here also chlorophyll A is there, which is 700. The reaction center, light energy more than 680 nm. Light energy is absorbed here to the surrounding accessory pigments. That light energy is passed on to reaction center P700. Again, P700 it releases the electron and it gets excited. This electron passes on to the primary electron acceptor, that is the peridoxine. So this peridoxine it passes on the electron into the lumen. So when it passes on the electron into the lumen, so that energy is sufficient to change NADP plus to NADPH. Okay? At the same time, two electrons are passed on, and that is sufficient to produce NADPH, NADP plus to NADPH. Okay? So it is moving here in the Z shape. Okay, so as we are left with just 1 minute, I'll stop here. So we'll continue in our next class. So quickly just switch on your video so that I can take a screenshot. All of you, quickly, we are left with just 1 minute. So today there was some power problem, that's why I could not connect the class as per schedule. We were late. So, so quickly just make videos on Chandi, Bharati, Rajshekhar, Tarak, Vishnu. Videos on Chandi so that I can take a screenshot for your attendance. So we were in a hurry due to shortage of time. Anyway, we'll discuss in detail regarding the PS1 photosystem 1, 2, that is the electron transport and proton translocation in our next class. Okay. So anybody wants to ask anything, you can ask. Yes. Vishnu, Tarak, Shyam Kumar, videos on Chandi, and stay safe. Don't go outside necessarily. If you go outside, wear mask. I'm happy all of you have attended. Most of you have attended. Keep it up and be regularly attending the classes. Rajshekhar, video off చేసుకో. So